We start with a very broad question, and what I would like to do is take a little bit a look into the crystal ball. What we experienced over the last 5 years in the field of lung cancer, and we've shown the slides here in different talks. So maybe to our colleagues from Heidelberg first, to Christof and Benedikt. If we look 5 years ahead from now, what we see in lung and some other cancers, how would you see, what do you believe will happen and how therapies will change, especially how these diagnostics tools we are discussing today, the wet lab part, but also over the big data, how will this influence the way we diagnose and treat patients in 5 years from now? Christof, please.
If I only knew. Well, we probably see this converging together. I think, for example, as one example, we have seen that there are now the things we're talking about today, early discovery, immunotherapy, and high throughput large data, where we are noticing that the patients with the highest mutation loads are in many cases, in fact, the ones that are most amenable to immunotherapy. So I think as we are growing experience, not all of our dreams will come true in how simple it will be to use it, but there will also, I think, be other uses of what we're currently discovering. So I think this is really still very much a discovery phase.
Yeah.
Yeah, if I may add to that, I think we will much more think in molecular categories, not so much in, well, still, of course, in organ-confined cancer entities, but we will subdivide into molecularly driven entities, like we've seen that in a number of cancer types, which are still nowadays be put into different subgroups. I guess we will hear something tomorrow on breast cancer about that. And not necessarily only with targeted therapies. We've seen also fantastic successes with immunotherapies, and I'm also convinced that we will have more successes with epigenetics therapies, because we've seen some cancers that have very, very few genetic alterations, but still they are cancers. They behave like the other cancers. They have the same molecular profiles with respect to gene expression profiles, and epigenetics therapies have the potential to transform the genome at many different places at the same time. So it's a kind of general genome-wide treatment, whereas a single interference with signaling has the potential to be effective on short term, but probably not on long term, because there are so many ways to get around that.
Thanks a lot. Any other comments to that? Yes, please.
I firmly believe that actually big data and what we're seeing today is going to be a game changer. We simply need to look at the metrics. We have so much progress, we have so much knowledge, we have new drugs. We do some simple maths. Let's take 5 medications, 5 biomarkers. Let's get for a classical clinical trial, we would need to actually staff more than 1,000 treatment arms. That's not possible. Now think about hundreds and thousands of biomarkers. Let's think about all the variants of unknown significance that today we cannot interpret because we haven't had the capacity to research them properly. And I'm pretty sure there is a ton of information hidden there. We can only solve these problems with a big data approach. If we only think about, let's take a few hundreds of biomarkers, a few hundreds of potential treatments, target treatments that we have, the human mankind, the few billions that we are on Earth are not enough to staff the clinical trial. In the end, what needs to change is the entire landscape, how we develop drugs and diagnostics and how we approve them. It's an interesting journey. We're just at the start, but I firmly believe it's going to be a game changer, and we see just the beginnings of it.
Thank you. From the audience, does anybody also want to contribute to this discussion, what the next 3 to 5 years maybe would look like in this field?
At this point, I wonder how can you integrate perhaps also sequences or of applications of drugs? That's because transcriptional response might lead to change which makes another drug more effective. So can you also integrate some sequences of drug applications?
Yep. So if I comment on that, of course, if this information is available, we can integrate that. There are also rational ways to at least produce hypotheses on how to do that. At the moment, I would not feel very confident to recommend this in a clinical setting, but we may want to try this out on patient-derived xenografts, for example, to see whether the paradigm would hold. However, I have to mention, to really test this in a clinical setting, and that's a general problem if you go, for example, to combination treatments, we will run into serious problems because there's a combinatorial explosion of the different combinations that you can try. If you think of different sequences of drugs, of different dosing schemes, and so on. And there's simply not enough cancer patients, even worldwide, to run these trials in a thorough manner that has been done before. So what we also need are new biostatistical concepts to make most efficiently use of what's available to actually and get the most use out of that. One way we've seen also is, of course, also to increase the number of patients in clinical trials, but even that would not be enough to run all the combinations and sequences that would need to be done.
Yeah, maybe I also would add to the previous question, just adding to what you just said, if this whole notion changes, and we definitely see investments around the world to really take advantage of the data that is coming out of the clinical context, out of the genomic analysis, and again, as this is more routinely than really captured, this knowledge base will build up, and we will increasingly leverage this knowledge base, as you mentioned before, to really understand, making more informed decision based on previous patient that has been treated. It might not be a perfect match, in all the depth that we see, but increasingly, if we all believe this notion of precision medicine is the right way to go, and clearly we have seen it is definitely helping us to provide better care, then those informed decisions will come. So clearly we see a lot of investment around the world happening in this context. Clusters are forming, and also the effort that we're doing with CancerLink, who are really reaching out to all the members to basically leverage also that data to build that one knowledge base, which then might also be the resource to help in those rare cases. We believe that this is coming.
Another thing that I'm pretty excited about is non-coding mutations, regulatory mutations. I think you mentioned, it was a bullet point in your second to last slide, and I was excited to hear about it, but it seems like the coding mutations, the efforts so far have, I won't say discovered all of them, but discovered the vast majority of them, but I think regulatory and non-coding mutations probably more degenerate in the numbers of different sequences that could have the same effect, and pretty much unstudied because there's that much more sequence space that needs to be explored. So I don't know what's going to happen with that, but quite possibly there are a lot of important drivers that haven't been discovered yet because we haven't been looking in the right places in the genome.
Right. Yeah. Christof? Yeah, I think some of the things that we are going to see as real advances are actually not going to be the ones that are super spectacular, big data, but the smaller size, like real-world outcomes. Do we really understand what the 83-year-old patient, what is happening to them down the line? Or are we just looking at 65-year-old patients with perfect renal function for the next 3 months or so, or until the outcome goal of the study has been met? I think as some of these projects really will afford us the access to data of the real world out there, I think also our treatments will improve. And that, I think, also will address the question, or will have to address the question of early discovery and prevention as well. So I think we will have to extend our range of actual data discovery, if you wish, in both directions on the time axis.
Thank you. I had another thought. When we discussed earlier about the tumor board and different ways in what we measure, and the discussions about proteomics coming in, epigenomics is coming back, I was wondering a little bit, if you look at drugs like PD-L1 and the tumor microenvironment and all what's going on within the tumor and the immune cells in the tumor, and maybe the question first to SAP, do you also look into things like digital ways to scan that and also bring this information in a kind of very robust way into into this report in the future, because I think that for those drugs, the DNA piece will be important, but still to look into the tumor, what's really ongoing in the tumor, and how the tumor cells fight the immune cells forth and back, and get that quantified and into the report might be also very important.
No, very important point, and I kind of touched upon this topic with this one piece of work which we did in proteomics, but here we are definitely relying on our scientific collaborations to really basically enable them so that they can use the technology. Basically, we take a three-tiered approach in our efforts. We have a research effort going on, we have a co-innovation effort, and we have more kind of standard development effort going on. And in the research part that we are driving, definitely proteomics plays a key role, and we have a number of collaborations ongoing, and if there are great projects here, happy to support them, because again, as you said, we definitely believe that in future there will be more capabilities that we have to empower in that area.
I mean, from the research efforts that Foundation Medicine is moving forward with, it seems clear that that's a big part of it. I'd say liquid biopsies and doing diagnostic testing out of blood is one, and then all the different applications of testing for the interaction with the immune system are the sort of two major focuses of research that we have going on. So we'll see where it goes, but certainly that's where we think it's heading.
Yeah, I mean, we know very well now that actually tumor cells are priming their environment like stromal cells to express factors that are suppressing immune answers and things like that. We need data. We are data people, at least in our company. As soon as we get access to these kind of data, we can model that. Actually, we have done efforts in this realm where we actually had, from model systems, expression data of stromal cells when they were regularly organized versus unorganized, and saw the difference between those. It's really fascinating. It's really basic research at the moment.
We're taking a large effort to collect the imaging data also on the immunohistochemistries and the other types of immunomonitoring assays as well. I think a lot of the data is just not currently available in a machine-readable format, or even something that a user can pull from various cases to quickly test a hypothesis thought or something of that kind. And we're obviously hoping that to develop discovery tools, availability of such things should also greatly improve.
Thank you. Another question now to Garrett. Garrett, in your talk, the first part of your talk was a lot about the technology. And I think it's interesting because we have a part of the panelists are really working on the right end, more on the bioinformatic part, while Foundation Medicine also has the whole wet lab flow. And I think the question for me, and I think that's also for many people, the question is how important is the exact flow you are doing? And I know that there's a lot of effort in that, or what would be kind of very robust quality control measures to combine different technologies, maybe also from other sequencing labs around the world, to make it suitable for good analysis in the end. Because we had the discussion earlier, if the sequencing is not done in a very good and robust way, you can interpret the data, but what does it really mean? Can you talk a little bit about what you think might be in compromise between just having only one floor or putting it together in a kind of robust system which could be also applied in other labs, which of course need to have a high standard?
Yeah, when we started this 5 years ago, I think it was quite clear that doing this at one center was the way to get it done with excellence. I think as the field moves forward, at some point that's not going to be required. I don't know when that will be. I think right now I would trust a group that had a lot of experience, but as other groups start doing this, if they can demonstrate that they have the same performance, there's no proprietary secrets. We've published our methods. A lot of them are derived from things that were developed in a research setting. So I don't think there's a fundamental limit. It's just a matter of accumulating the experience and the technical know-how all in one place. I mean, our company is 400 people devoted to just doing this, and I think a group of 10 or 20 people, it's going to be hard to have the same attention to detail and the same amount of resources applied to it, but inevitably that is how it's going to go. So what
would be the most important quality measures? For example, if you purify the DNA, you know, out of the tumor cells, or later on, before or after sequencing, what do you think the 2-3 steps which are really key, basically, to guarantee for the quality?
So certainly getting high coverage. We're going for over 500x coverage across the whole target region in order to identify mutations in tumors that are 10 or 20% tumor nuclei is critical. And then I think validation is really the key. It's not that any one step is so critical. It's that from the start to the finish that you can demonstrate that the test is working. And so I guess, I mean, I also highlighted removal of PCR duplicates as critical. There's a few different ways to do that. That's not it's not a secret, but a lot of groups aren't doing that now. So,
yeah. Maybe Christof, your experiences here in Heidelberg with sequencing?
Well, one of the questions that I have, obviously, and something that we've been fighting here, is the question of sort of format and availability. And we have seen basically 3 commercial contenders in this space today. Obviously, the model is changing in that sense that the data, some of the data systems are open or even agnostic to previous formats. We've heard a lot, I think, about adjusting data quality. And so one of our primary concerns here at the center, but I think also going forward for the entire space, is, are these solutions going to be, can they work together? Can the scientific discovery access the databases that are created, which are obviously already humongous in some cases? And how is that going to play with the interaction between academia and these obviously very important parts of the industry?
Questions from or comments from the audience? I think I see an arm over there.
So I'm from the US, so thinking about cost effectiveness.
Mike is coming.
Thank you. So I have a question about cost effectiveness. So clearly the effectiveness, I guess you've teased us with, but the cost we haven't discussed at all. Do you feel like it's going to, in the end, because of precision medicine, we're not going to be wasting ineffective drugs on people who wouldn't benefit from them? But the cost of what you're talking about is really something that in the US we're trying to think hard about, in other places. Maybe not very effectively in the US, but we're thinking hard about it.
Does anybody want to take this question first?
Happy to take the question. So maybe also quickly to the previous question. And this also goes to the cost, at least from an IT perspective. So openness is key, right? We need to be able to integrate efforts and make sure that we can work with different efforts that are underway. We'll see this will be standardized. This will become across the globe. We'll see different efforts coming up, not just one excellent lab doing this. So in that regard, we definitely see this happen. That is the perfect lead and also into the cost discussion. If we listen to Michael Porter and what he's proposing, especially in the US, how we can deal with the rising costs in health. Clearly, what he's suggesting is to create networks and make sure that in those networks there's the right center of excellence that deals with certain parts of the procedure but basically they cannot and should not cover it all. There should be maybe some of the specialized treatments should be done at a center like here in the University Hospital in Heidelberg. And as long as you pointed out afterwards follow-up treatments in the community, then it's done still with the right level of documentation. And then the focus is on patient outcomes as it relates to cost. We firmly believe that this is the right way to go. This doesn't ultimately address the questions of very high costs for chemotherapeutic treatments, especially innovative ones, the immunotherapy therapies right now. But again, this is maybe a singular point that we see at this point in time that will change over time. But clearly this notion of treating patients network with specialized, personalized treatments, so that we turn, cancer into a chronic disease, maybe even with right preventive measures up front. Again, I believe it's consensus right now that this is the way to go, and certainly this is what we are supporting from our perspective.
I think, of course, measuring effectiveness is one key, but we're still in a discovery phase. So really what we have to do is to think as hard as we can about which molecule in an optimal case to use for the optimal treatment. The price of the charge for that medication should not be a preoccupation of the physician researcher at this point. And I'm getting a bit upset if sort of the cancer cause, which is number 5, I think, in our health expenditure in Germany, where I think around 15% of the total, with something that is very important and obviously accepts the lives in so important ways of so many people. It gets me a bit upset if I think that we, on the research side, should sort of censoring our thoughts based on what things might cost in the future. I'm completely and entirely and utterly against even thinking that with regards to that. Now, that said, of course, what companies do in terms of charging, the optimal rate for their recovery, I think has to be insightful and has to have a systemic thought on how that can actually work in society and the system as a whole. But I don't think that is something that researchers should be preoccupied with. This is a political question that is, I would see that completely different from what we should think.
It's not key for you, it's absolute key for us. Yeah. I mean, cost effectiveness actually is absolutely key to adoption and broad adoption. And who ensures cost effectiveness is the one who pays. In the end, you want to get reimbursement for your test. You can only be reimbursed if you prove actually cost effectiveness of your test. And that directly comes to the cost. And that directly also comes to your previous question about sequencing technology, at least in my view. So I firmly believe, I mean, today's bottleneck is still sequencing. We have to wait for the next wave of technology to make that much cheaper and more reliable. A lot of the problems that we are trying to solve algorithmically today will become obsolete with next generation technology. There's a huge promise in the nanopore and electronic detection. Let's see where that carries us. I believe that the technology change also on the signal generation side holds the promise to make that much cheaper and make it much easier to bring that broad adoption and get the cost effectiveness actually up.
Maybe if I just can add one question to that. So if everybody says, okay, sequencing should get cheaper or much cheaper still, I mean, how much of the cost is really the sequencing device versus the whole workflow and the quality control steps and all of that, which needs to be included? I think we've discussed that. So is it really realistic that even so if we have a next generation sequencing again, that the costs will really drop by 50% for the whole procedure or so?
I believe so, because a lot of the complexity comes actually from the complexity of the test. I mean, today we need complex chemistry. This is still complex chemistry. We have optical detection methods. We need to transform all of that. Just think about electronic detection here, and you don't need to shredder your DNA into small pieces and then reassemble it later on. I mean, a lot of the steps that today we have just for procedural reasons will become obsolete. I firmly believe that it's going to become cheaper also from a process perspective, but maybe you're more the expert to talk to that.
Sure. I mean, I'm not in the financial group at our company. If you could just take DNA out of a tumor, do no lab process, and just get the sequence out of it very easily, that would, I'd say maybe a third of our company does that part of the process. So it would make it, I think what will happen, though, probably is as soon as diagnostic testing gets cheaper, you will do it more frequently. So my instinct is that the cost of the diagnostic tests are really a very large part of the cost of treating cancer patients. And I'm glad I don't have to make the decision of how much is it worth to keep patients alive for 4 months on average. You know, that's a healthcare economics question. So.
Yeah, also see, of course, keeping in mind that we're talking about things like liquid biopsy, early detection, and other methods where we are trying to collect more and more of that data and use more, and if we also have to, more expensive diagnosis to detect disease earlier or avoid it altogether. So the single procedure may get cheaper, but I think we are going to end up spending up a whole lot more on diagnostic procedures of all kinds.
Thanks. There's a question up there.
Yeah, hi. Simon Valentine, Illumina. Just as a point of note, I actually happen to disagree with you, Stephan. I think that the cost is very much heavily weighted towards bioinformatics and data interpretation than it is generating the sequence data in the first place. But my question actually relates more to do with how the panel sees us being able to resolve issues around consent and patient data privacy in terms of us being able to take advantage of big data analytics, big medical data, because that's another bottleneck that I think we need to think about as a society and as an industry.
Maybe first let me disagree with your disagreement. Because I think a lot of the bioinformatics tasks that we have to do, at least in the beginning of the analytical pipeline, has to do with the complexity of the data as it's produced by today's current technology. But we can discuss that afterwards. Actually, your question is also my question. How can we tap into this data and resolve the problems around data privacy? And I think in this country here, in Germany, it's even more a problem than it is in the US. Here we have, I mean, we can use the data only according to the initial purpose for which it was generated. Every other secondary use, and for every single question, there would be actually the requirement to get approval for that. So now imagine you have a real-time system, you're asking 10 questions in a minute, getting approval for every question, it's not possible. I mean, that's why I had that slide with the bottle in the end of my presentation. I don't know how to get this data out of the system. It's sealed within.
Can you ask for that approval in advance from the patients?
Yeah, there are ways to do that in academic collaborations. There are ways around that, and this is being done. Many things are in the kind of a gray zone. I would like to have that really a broad and open discussion, so that all patients can benefit from that. Of course, I understand this is the most private data that you can generate. It's your genome, but we somehow got to resolve that.
And I would echo that, and thank you also for raising that challenge, Simon. I mean, clearly, especially here, as you already pointed out, Stephan, this is a challenge which we are facing. But I believe that as we show the power of and the benefit of what we can do, it's almost like with every other new technology, whether we're talking about the introduction of the first machines, whatever, big concerns in the society, whatever that will cause. I'm hoping that we'll have a public discussion about this topic increasingly, so that patients increasingly will give their data to trusted organizations such as the NCT, such as the DKFZ, and they will basically use the benefit they will use the data for the benefit of the patients. If not the patient directly impacted, at least to really advance research and have the next patient benefit. And we see some of that.
So final comment.
Just, I mean, from a perspective of somebody doing data analytics, it's of course very nice to have data available from large data sharing all across continents and so on. But I want to stress that actually data privacy is a key issue here. as long as there is a system that doesn't allow to get out the data of an individual patient, but just aggregate information and make this aggregate information in a way that you cannot trace it back to an individual patient, and there are methods to do that, and this system is very heavily protected, it should still be okay. But still, we should respect data privacy because, as has been said, the genome is the most personal part of ourselves. And there are ideas around that in the future. Maybe you will use your genome sequence to identify yourself for certain internet applications. And just imagine somebody steals away this information. So you can change your credit card number, but you cannot change your genome. Yeah? So you cannot prevent that anybody who has this information will do something with that in the future. So I think we should take this seriously, and there are ways to do it. It may cost a little bit more than was anticipated, but it should be done.
Right. Christof?
Yeah, I think it has already been said. Some of the things that we are discussing about data protection, also that our current medical practice is data protective, is actually embellishing the model that we're actually losing a lot of the data that we actually should be using. I think eventually the quality of our findings will define what the patients, what the citizens, what the customers will be willing to give us.
Right. Great closing remark. Thank you all. Thanks a lot for this discussion, and thanks to all of you for staying. We have half an hour break, and I think we are around. If there are more questions, we can easily continue in the break. Thanks a lot.